Now, the process of meiosis is a type of cell division that divides a cell into four daughter cells. And those cells are called haploid cells because in the end, those cells will not have this exact number of chromosomes as the parent. And also, those cells that are produced in meiosis are genetically different from the parent. And this is the result of different processes that happen as this process occurs. Now, what are the stages involved in this meiosis process? So meiosis process has got two types of processes inside it, which we call as meiosis I and meiosis II. In meiosis I, we'll have the same stages as in prophase, we'll have the same stages as in mitosis, and also in meiosis II, we'll also have the same stages. So basically, the stages are summarized in the mnemonic PMAT: prophase, metaphase, anaphase, and telophase. Now let us begin with the interphase stage. We know that before a cell can start undergoing division, it must enter a resting stage, which we call as the interphase. In the interphase stage, the cell is not dividing, and hence it is not included as the part of the stages of this meiosis. It is just a stage in the process of cell division. Now, during the interphase stage, the cell is said to be at rest because it is not dividing. The reasons are simple. This is because its chromosomes are long and thin and hence cannot be seen under a microscope. In this process, that is where also a cell replicates its chromosomes. Because remember, for a cell to divide, each cell must have at least some copy of the DNA. So it must first replicate or produce exact copies of itself so that once it undergoes the process of cell division, you can have two cells which will have genetic information which is full. So let us begin with the first stage of meiosis, which we call as meiosis I. In meiosis I, the first stage is called prophase I. And in prophase I, what happens is exactly or very similar to what happens in prophase of mitosis, where the nuclear envelope breaks. This nuclear envelope breaks so that the chromosomes inside the nucleus can be allowed into the cytoplasm so that cell division can now start taking place. Aside from that, another key point that happens in prophase is that the centrioles, which are responsible for forming spindle fibers, you know that spindle fibers are the ones that help in separating that chromosome strand. So those centrioles begin to move to opposite ends of the pores to form spindle fibers, which will help in separating the chromosomes. Aside from that, in this process of prophase, the chromosomes now start to condense. As they condense, they become short and thick, which allows for them to be viewed under a light microscope. And so cell division now starts taking place inside the prophase I stage. In the prophase I stage, that is where what we call synapsis takes place. Synapsis is a situation where a pair of homologous chromosomes, not really, sometimes it's not really homologous, but a pair of chromosomes mix up at a point which we call as the chiasma or the chiasmata. So when they mix up at that point, what happens is that those two pairs of chromosomes separate or exchange materials. And that is the first reason why the cells that will be produced under meiosis will be genetically different from the parent, because there is exchange of genetic material during genetic recombination, which we call as crossing over. So when synapsis happens, when the chromosomes come together, that is called synapsis. And then the sharing of information through the chiasmata, we call it as the crossing over. So there are more stages under the prophase, but um, all you need to know in general is this process of prophase one happens in that type of situation. So those are the key points: crossing over, synapses, and the chiasmata. From there, we now move to the metaphase one stage. Anytime you hear metaphase, metaphase means the equatorial plate or the equator or the metaphase plate where these chromosomes have to align. So these pairs of chromosomes will start aligning on the equator. Another point to keep in mind before we even move to this metaphase one is that in prophase one, we have a pair of similar chromosomes pairing together. So you have a chromosome and another chromosome pairing together, and that is called a bivalent because we have a pair of two chromosomes assembling together at one point. So in metaphase stage, these pairs of chromosomes now align at the equator. Remember, in metaphase one, there's only alignment at the equator and nothing more happens there. Then we move to anaphase. In the anaphase stage, those chromosomes now begin to move to opposite ends of the pores, or they are separated by the spindle fibers. Please note that during the meiosis I stage, the chromosome number does not reduce because what separates are the chromosomes and not the chromatides themselves. Chromatide is one part of that chromosome, and two of them makes up a chromosome. Then in the prophase stage, what you would expect is normally the same as in prophase 1. Of course, you have the nuclear envelope coming together again because you formed two cells which are genetically identical to the other. Of course, they'll have some differences due to the crossing over, but in this situation, they haven't really separated into their chromatides. Then what happens is that meiosis II now begins after the cells have divided into two and having a chromosome number, right? Then in prophase 2 now, these chromosomes will not replicate as they should have done during an interphase stage. What will now happen is that they will now become short and thin because remember, during the telophase 1, they became long and thick, thinking that the process had ended. So in meiosis 2, those chromosomes now become short and thick, getting ready for a second round of cell division. Then, in metaphase, as you would guess, those chromosomes align at the equatorial plate again. And then, during the anaphase stage, those chromosomes now separate into chromatides by the spindle fibers and begin to move to opposite ends of the pores. And then finally, in the telophase stage, you have the nuclear envelope, the nuclear membrane, starting to divide in a process called cytokinesis, which will divide the cell into four. Each will have a chromatide which will be genetically different from the parent, and as you can see, it will reduce this number to by half because these produce haploid number of chromosomes compared to the process of mitosis. So how is this process very important to organisms? Firstly, it helps formation of gametes. During fertilization, they are restored, and so it is very important for these gametes to contain half number of chromosomes to avoid a situation whereby the organisms will have more chromosomes after the process of fertilization and forming a new individual, right? Yes. To avoid processes or complications like Down syndrome and so on. And aside from that, this type of division meiosis is the basis of sexual reproduction. And so, knowing that, I'm sure you are able to now relate exactly to different type of questions and how it comes. So basically, that's how the process of meiosis happens. Thank you.